case I mixed up. But if we were to take a leaf falling off of this sugar maple out here, Acer rubrum, and it's falling into, let's just say, a pond, okay, something moist, like our bodies. What happens in the decomposition of materials this time of year? This is the big nutrient flux. Wow. Okay, everything's falling down. You're smelling the geosmins from the bacteria from excuse me, different breakdown products. We have all this stuff going on. If a leaf is falling off, the things that fall out of the leaf first are the soluble materials, like tea. Like taking your jasmine tea and throwing it in hot water, it turns some kind of greenish, blackish color. You enjoy the aroma. Aroma is oils. They're somewhat non-soluble, but they're broken down in the environment. But then the sugars and the proteins come out first. After that, as things get more recalcitrant, we talked about what recalcitrant is, they break down slower and slower. The next things to break down are some of the polysaccharides. DNA breaks down, but there's just not much of it. So a little phosphorus gets trickled in there. Okay, that's cool. Okay, what happens next? Okay, the ones that most people know about are cellulose. But cellulose, it takes a group of bacteria to break down cellulose. I don't break down cellulose. That doesn't help me in my question about the large intestine. Okay, hemicellulose, easier to break down. Okay, got a few more sugars in there. What about suberin? What is suberin? Suberin's like a polyester. It's the great polyester leisure suit of the leaves. Okay, and then you get to really recalcitrant stuff like lignin and tannins, kind of stuff you may enjoy also in your tea, perhaps in wine. Things that will break down very slowly, and we know this to some degree when we age wine or we have certain really nice different teas, like you might pick up in Chinatown or something where tea is thought of very differently, perhaps as the Americans do, at least in my experience. These tannins break down, give different characteristics. That's a leaf. Now your body is chewing leaves. You're chewing whatever, hamburgers, or if you're vegetarian, fruits and vegetables and nuts. Okay, it's making it through your mouth. It's getting all wet and moist and a little bit broken down in your stomach. It made it through the intestines. Okay, why does the large intestine now have the highest amount of species? Different substrates. Okay, one thing that we keep asking about in this class is species diversity. Why is there so many species? How can there be so much diversity on Earth? Okay, even in your body, even though you might be able to break down all your foods into the four big biomolecules, that's true, proteins, polysaccharides, nucleic acids, and fats. Now your body is broken down into different subcomponents. Some things are indigestible by you. Cellulose, really you don't really make a dent in cellulose. I'll put that out. So that's your fiber, right? But the other things that are breaking down have been broken down either mechanically or chemically, then enzymatically, and now you have all these breakdown products in this anaerobic area. This is like going into the middle of the swamp and watching the leaves decompose there, okay, in nature. Kind of smelly. Yeah, we know it's kind of smelly down there. Okay, so we got some sulfide being given off. Okay, but then we not only just have these very simple oxidized components, but you have anaerobic decomposition going on. So you have a lot of fatty acids. You have a lot of partial breakdown products. And now, all the species that are only working at kind of a marginal anaerobic respiration or facultative respiration type of situation are all taking their little piece of it. It's going by in a slow river that's moving out of your body every day. Okay, and as this river of sludge is moving through your colon, these guys are taking a little piece of those breakdown products that are going on. Everyone taking a little piece, dropping out a break-off product. That breakdown product is being picked up by something else. If your colon was aerobic, it would burn it up much faster. The species would be much shorter. That's not the gig. Okay, that's not what's going on. So, as we ask that question of diversity, why are there so many things? You have to think about energetics, habitat. You have to think about what the substrates are, and then who can get a piece of it. Here we're talking about fermentation. Are you guys okay with that, or was that like, oh, I don't know what the hell he's talking about? Yeah, think, you know, use all that great stuff you're learning. And you're learning organic chemistry, and you're learning, you know, about forests and physiology and all this stuff, and it all comes into play here. The chemistry, the biology, all those rules that work for the rabbits and the turtles also work here, except that they're just not very creative about their metabolism. These guys have a few other tricks up their sleeve, these guys being the bacteria. All right, let's get back on track then. So we've kind of painted an idea of what's in the enteric bacteria. You know, we know that it's anaerobic. In your duodenum is what? Is that Latin? Duodenum? Is that? I know it means 12. I don't know what language duodenum is. Anyway?
It's Latin.
Is it Latin? Okay, so at first, you know, that's your first 12 inches of your intestine, as I understand it, descending. Okay, by the time you get the end of that first 12 inches of that, whatever it is, several meters of tube, you're anaerobic, but your pH in your body from the chyme has been brought back up to around neutral. Now, is it going to stay that way? To some extent, you have some buffering capacities going on, okay? But generally, it's going to tend to start to turn a little bit acidic. Why? Fermentation. Okay, now some fermentation will yield acid, and the mixed acid fermenters are a part of these enteric bacteria. Okay, there's a lot of stuff in there. All of a sudden, you're just not going to go back to pH 2. I mean, there's a lot of things competing in there. But anytime you have these reducing sugars, that group will tend to put out a little bit more acid. Anybody in lab who we did fermentation tubes with E. coli, bang, it went from red, phenol red, to yellow. You know, there it is, okay? There's your indicator. If you had a pH meter, it'd tell you the same thing. Okay? So this fermentation of sugars is really important, and it's there. Is everything in your intestine sugars? No, but a lot of our stuff is. If you look at what you're eating, if you're eating the 4 food groups, you know, a lot of that is carbohydrate, and so fermentation of sugars, directly or indirectly, are in there. I think we've been through this. Oxidase negative. It's not that they can't work aerobically, but that oxidase test really works well for obligate aerobes like Pseudomonas, which we'll come up with. So, you know, I think I was reading on one of the tests. My life of correcting all you guys. I can see your faces, but the words blur together. But someone said, well, you know, the oxidase. I think it's just bored with me. Yeah, okay, I'll get back. But they said the oxidase test says that you know it's negative, therefore this thing's an obligate anaerobe. You got to be careful about that when you deal with the guys that are working in the middle. Is all I'm trying to say. You know, you're right to be saying. Yeah, I can't tell you. I got to spit out my gum. Yeah. It's partially the name of my dog. Okay. Okay. So, you know, it's true, you know, that the oxidase is associated with the aerobic cytochrome system. That's it. It's just a matter of this particular step goes away in these guys. So that's the deal there. So you can't just say, well, E. coli never does aerobic respiration. We know that that's not the case. Okay. Yeah. We also know that some members of these groups, like E. coli, to get by without oxygen, they don't go straight into fermentation. They might need to do anaerobic respiration and can reduce nitrate, which is kind of cool. You know, they have a lot of flexibility. All right, I think we went through this. I should probably pick up some speed here because we got a late start. Please come back. Okay, it was a slow start. I'll pick up. Okay. We kind of know this, but you know when you see those enterostrips, like all those different tests we do in lab. If you're not in lab, we do like 20 different tests. We do the most when it comes to the enteric bacteria. Even that is reduced because I've picked, I've kind of cherry-picked the species for you to work with, but, there's a lot more enterics. You can put all that type of work into a little strip, and every one of those little biochemical tests are like the size of a teardrop. You inoculate it, and you can get the reading in 24 hours. You can score it like a report card, and that is very helpful. But still, you got to do like 20 different tests. So they're very closely related, and they all spit out and do a little bit different of an enzyme thing, but we can do rapid diagnostics. It doesn't mean that your time in lab has been wasted. All those tests are still part of that type of typing if you do it with culturing. And it also teaches about physiology. Okay, I think this is where we left it. We have two main categories, the mixed acid fermenters, and then this compound, butane-diol. That's just the way it breaks down, okay? That's it. The butane-diol is one group. It's from a pathway of fermentation. Yeah, do you get some CO2 and some acid? Yeah. Okay, it's still fermentation. But it's not to the extent that you get with the mixed acid fermenters. You go, well, how much is enough? It's enough where you can, if you look for this precursor to the 2,3-butane-diol, I know in the lab we mentioned this, that is something we can measure easily. And this is a little bit harder. You can get a color reaction here. If you get this, then it therefore is not this. And so we do tests that measure both acid and a positive negative for acetone. Oh, got it. That's it. I got the news surrounding that. Okay, so this is just showing a little more chemistry to it. The main thing in on this one to talk about is the gas formation. Hard to see in here, but we got a little bitty tube in there. It's collecting gas. These things are coming out, so we see a bubble. And that bubble is a high amount of CO2 and hydrogen, about in equal proportions. And then there's acid. A good one, your poster child here, E. coli. Great mixed acid. With the butane-diol, we'll talk about a few of them, but Enterobacter, that's a genus and species, Enterobacter aerogenes, worth knowing in this class. I think that you'll be fine remembering that. You'll not get appreciable gas in here. I know it's just innocuous, but even in here you'll not get gas, and then you're seeing a color reaction that they're using to look for the presence of butane-diol, because it's not all converted right away. Some's in the acetone still position, and then you can detect that. All right. Today is not my day for audiovisual. Okay, a little bit about E. coli. I mean, there's a lot. Now, where does E. coli live in you? Does E. coli live in you? Do you have E. coli in you? Yeah. Okay. Yeah. Where is it? Here. Here. Intestines. Yeah, it's Enterobacter. Okay. Now, we tend to think, and you can ask perhaps people who you think may be quite knowledgeable. They tend to think, yeah, we have bacteria, most of it's E. coli. No. Okay, probably less than 1% by biomass. By species, it's just one on a big chalk list of names. Okay, now, why do we make such a big deal about E. coli? Because E. coli was easily culturable. It can do so much. We can culture it a multitude of different ways, and we started taking it apart first. The molecular engineering that was developed. E. coli was the white rat of the bacterial world. The cloning that we talked about a few weeks ago. Cloning, we make a surrogate population with E. coli. Look at E. coli, E. coli, E. coli. That's the deal. So naturally, if E. coli is the only bacteria you know of, you're going to think that there's a lot of it. You tend to do that. You now all know better. Oh, not so fast there, my friend. It's less than 1%, yet it does some cool stuff for us. We know where it's found. We get some vitamin stuff from its fermentation. That's kind of cool. Thank you, E. coli. Not dominant. If oxygen is present in the first part of the intestine, it will take it away, as will any of the other heterotrophs. Okay, that's it. Yeah, you can swallow oxygen. You have it in your food, but if it's there, those heterotrophs, they're gonna snuff that oxygen right out. They love that. Okay, you would have big problems if you started piping oxygen into your intestines. Your bacteria would probably start heating you up to a point. You wouldn't ignite, but you would heat up like that. Okay, what we got here? So E. coli. Several things now of note. So wake up if I put you to sleep. Diarrhea, yeah, traveler's diarrhea from E. coli is easy to get. It's not fatal. You get the runs, you get diarrhea. It makes massive, either it stops absorption or dumps, depending on massive fluid into the lumen of the intestine, and of course that makes everything real liquidy and hard to control. Okay, so that comes up a lot. When you're traveling, eat it, peel it, or leave it. A good thing to live by, don't always need to do that. This day and age, things change all the time. Be careful of ice cubes. You know, if you're traveling in a warm area, everyone likes to have a cold soda pop or something, or a cold glass of water. The one thing, you know, people will always say, oh yeah, I only want to drink water that's bottled. This day and age, that's easy. Make sure they're not recycling bottles in some places if you're in a questionable area. Okay, so you snap that, oh yeah, would you like some ice? Oh yeah, you bet. Bam, bam. Okay, all of a sudden you're on the head, right? You're on the toilet and not feeling so good. Okay, coli can be a problem in healthcare, amongst other things. Things tend to get mixed up between anus and the urinary tract. So it's very common to have infection. Again, more dangerous with people whose immune systems are starting to degenerate, like mine. Less threatening for younger people who have a good immune system going on. Here's the next thing, enterotoxins. I want to talk about enterotoxins for a bit. And so we have some categories here to sort out if you haven't gone through it before. An exotoxin, exo, right, goes away. A toxin that is released extracellularly, so exotoxin, something that spits out. Not all bacteria do, some do. So I'm going to kind of cover a few of these categories. Okay, next ones we have are the enterotoxins. Okay, entero, you can think of enteric, that's pretty simple, you know, a good association. Acts on the small intestine. Massive secretion of fluid and lumen, diarrhea, we've covered that. You know, there's other, we know of other toxins that can occur, cytotoxins, to cells, cyto, tissue. Neuro, of course, obvious there. All right, the endotoxins are a gram-negative bacteria association. In a way, it's not really a toxin that's being let out of the cell as much as it's an interaction, but it creates a toxic effect. Okay, gram-negative bacteria-associated toxin. So here we have to go to that outer membrane area, the lipopolysaccharide, and go and we look at the O-specific antigen response of that outer membrane to the tissue. Not all bacteria do this. You have a lot of things that are associated with different parts of your body, fine. Sometimes just different strains or different bacteria can make this association. You have E. coli in you. Most of us aren't experiencing diarrhea this morning. Maybe some of you are. It's not all that uncommon, but it's a matter also of how much will elicit a response. So, yeah, talk about that inflammatory response in animals. Now, in some cases, the toxins can be quite dramatic. I haven't heard of a case of E. coli 0157H7 recently. Has anyone ever heard about it in their life? Because you're a whole different generation. Well, then let me explain briefly. It wasn't all that long ago. I don't know if I have a date up here or not, but this strain of E. coli showed up. You know, it's not one that's normally a part of you. Those call letters talk about the protein. You gotta tape that's the second one. You gotta tape that to your fingers. But they're talking about the components of the other membrane. Kind of like when you see H1N1 stuff and viruses, they're talking about certain components there. All right, this particular strain showed up in much of the same type of pathways we see almost every summer or from time to time in the media, and that is either by the fecal oral route or from the contaminated food, contaminated water route. Okay, for instance, undercooked hamburger. Okay, please excuse me if you're a vegetarian. I'm not going to dwell on meat eating, but I'm going to gotta make a point here. If I eat meat. So if I want to eat a steak that's rare, why am I less at risk than if I want to eat a rare or a medium rare hamburger? Pretty simple question, but let's think about it for a second. Please say it again. I just couldn't hear you.
Because only the outside of the steak is exposed.
Yeah, the steak, I mean, if you're eating tissue of any kind, I don't care, vegetable, whatever, if you got bacteria in that tissue, you should be really thinking about it, or that muscle mass. Okay, so, given that it's a major muscle of a cow or something, you sear that outside, the part that's probably been in contact with whatever, butchery of some sort, you have pretty much taken out the risk factor, and the rest of it's up to you how you like to chew on things and so forth. Why is hamburger the problem? Well, hamburger is the opposite. We're taking meat product, okay, oftentimes mixing with pork, more times to get more money, mixing with water, which they do a lot now, to jack the weight up. And then it's being all mixed together. So even if you have just a little part of the hamburger that's contaminated, bacteria are small. You're mixing that stuff all together and grinding it through, some kind of meat grinder or something. You're really, really inoculating. So now just searing the outside and leaving that inside, rare and a little bit pink because that tastes better for some people, or they just prefer that. Now it's a different risk. Now you're putting your risk factor out of the line of control, which would be fire or heat, and putting it into the hands of your food worker. Oh, I really hope that everyone who's touched that piece of hamburger from here to Nebraska, has been clean and done a good job. In this day and age, a pretty good assumption. Every once in a while, it comes down and nails it. We see it more times like in the summer. Hamburger gets left out, or in food processing, people are very good. They don't want to lose their jobs, and they don't want to lose millions of dollars recalling hamburger, but so they're cleaning these machines. But let's face it, your degree of error or safety is marginal. Error is maximized in summer in some of these conditions, depending upon refrigeration and so forth. So hamburger, yeah, I mean, not a big risk factor, but you can get sick from hamburger pretty easily. So, today we'll find out how many thousands of bacteria live in your hamburger before you cook it. Some people started eating only vegetables after that. Contains, so here we go, we have this plasmid that creates this toxins released in 157, and the upshot from this, you don't have this strain in you. You're carrying disease. You're carrying stuff that maybe, and I hope to goodness that you never expressed in you, but some of you are carrying tuberculosis, you're carrying typhoid, different things, but you'll probably never express. If you had this in you, this toxin made your blood cells leaky. Okay, basically you would have bleeding going on in the intestine. If left untreated, it would take apart your major vascular organs. So yeah, the kidney, imagine the liver's right behind it, would become vastly eroded. And almost to the point, that scary stuff you hear about Ebola where blood is coming out of people's eyes. It's not quite like that, but kind of. And this is, of course, E. coli. So what's going on here? Okay, this strain emerged. Not quite sure where this plasmid came from. I don't. Maybe the CDC knows. I don't. This plasmid emerges. It's transferred around. It creates this effect. I think one of the most tragic ones was in a swimming pool. There was a kid that was infected, didn't know it, and they're swimming. What, I don't know, just to be practical, kids in swimming pools, what happens? A lot. They get very relaxed, right? Also, if you're not feeling good, it's also a problem. Sorry to be practical this morning, but that's the case. This one child not feeling well released this, and there was an outbreak amongst these kids. Now, the problem with hemorrhagic colitis and also the failing of the kidneys, yeah, this will respond to antibiotics. E. coli, we can nail E. coli, not a problem. The thing is it happens fast. With kids' metabolism, it happens really fast. Can't cut it out. You can respond to antibiotics, so some of the kids negatively respond to it and knock them out. Is this one? Anyway, that's the thing about no time. Yeah, you try to cut stuff out. Yeah, other incidences. And this is 1996. Maybe that's. Were any of you guys alive in '96? What were you doing in '96?
Being 1 year old.
1 year old. Well, back when I was your age. Okay. Well, anyway, okay. History, but let's we learn from it. There you go. Okay, it's like talking about toxic shock syndrome. I mean, I remember vividly in the 70s when that showed up and people were getting sick from tampons, and now you mention go, yeah, I kind of heard of that, but sometimes shows up sometime, not even a thing. We learn from it. People changed habits and manufacturers changed what they did. Here we think a little bit more about food. Okay. Salmonella. Okay. Salmonella, probably one of the best ways to get sick is to get salmonella food poisoning. Very contagious, very easy to get. I think the biggest thing I hear from my wife all the time is I should be more careful preparing chicken because we're all going to die from salmonella. Is that what you? Well, you're not. You're very married to my wife. I mean, do you hear that salmonella is like this chicken disease? Yeah. The skin. We can do skin in lab too. I have some chicken. Skin is amazing what lives on chicken skin in the refrigerator. Okay, so you don't have to know these stats. It just is, there's a background level of salmonella food poisoning shows up for all these different reasons. About 1/3 of food poisoning comes from restaurants. Not that restaurants want to make you sick. There's just a lot of moving parts going on. And sometimes there's stretching menus, all kinds of things. If you work in a restaurant, there's all kinds of ways that things can go wrong. Most places do it right and don't hurt us. This is just talking about where we're looking in the outer membrane for these types of toxic responses. Salmonella is amazing. There's 1,000 different serotypes. If you look it up in the Burgies, it's mind-boggling. Okay, go have an argument with Dr. Bonnicum. Not hard, but the thing is that he'll come down. He'll say serotypes are not individual different organisms. Okay, if it's not defined on the species level, it doesn't really exist. Try telling that to someone who just is losing their guts to diarrhea from a salmonella serotype. We go back and forth on it. I've talked to other people about it. So this is the same species. Yes, the DNA, yes, it's the same species. But the variation within these are called serotypes because of the response in vivo that will make you sick or not sick in different ways. Some salmonella is fairly good. I pulled salmonella from your use because it's getting scarier and scarier. Gastroenteritis, okay, endotoxin, I think we covered all this. I get ahead of myself. Probably get sick from salmonella, it's not hard. Okay. The fecal oral route is easy for salmonella to some extent Enterobacter. Anyone remember this restaurant called Chi-Chi's? Is a Mexican restaurant? Okay, well, I think they're out of business, but not because of this incident. Chi-Chi's, a few years ago, all of a sudden, like Taco Bell, okay, popular Mexican type food.
I think they still make salsa.
Yeah, you still see the salsa around. I
They
don't
still
think
make
this
the
story
salsa,
will
but
affect that too much. The thing, I think you cook salsa. But what happened is a lot of people started getting sick all in like 1 August of a year, some years back. And they narrowed it down to the lettuce that they're sprinkling on tacos. It's Mexican food. Here's some lettuce, here's this, it all looks the same, tastes about the same. There you go. And so, lettuce, oh, you can't cook lettuce. So now we're back at to that, now we gotta find out, the chain of command, who had it, what was going on. Okay, they said, well, did you wash lettuce? Yes, we make our lettuce, it's washed, it comes clean. Oh, well, that seems reasonable, we're trying to do a good job, we don't wanna make our people sick or they won't come to our restaurant. They went back, this lettuce was grown in the Southwest in this case, okay? And in the Southwest, in the summer, pretty hot place, so there's irrigation. They looked at the irrigation waters, like, well, it's coming from a good source, what's the problem? Cattle in the field, periodic rainstorm, or cattle just walking into the irrigation ditches. We're doing what cattle do at certain periods, just, okay, there it goes. And they're rinsing the lettuce with what looked to them to be clean irrigation water, and that translated into food poisoning across, actually, the country as the food disseminated. Every once in a while, you'll see that lettuce thing that comes up, and it crops up easily. Again, when you travel, I'm sure I told you the story on the Brazil trip of 23 scientists got deathly sick from, not deathly, very sick from diarrhea. One got amoeba dysentery, yes, pooping blood from eating the food and stuff. They're washing, they're thawing the meat from the market in Amazonian water. That was being cooked, but that was still a little bit gross for me. The Port of Manaus is gross. But then also washing the lettuce and stuff. I love salads, but I wasn't eating in those days. So 3 of us did not get sick. I think it was just luck, actually, but most of my friends were sick. But it's something you just don't think about. But you look outside, look at the Amazons, like, I'm not gonna drink that. Maybe if I was brought up here, but as a white guy from Illinois, I'm gonna drink Amazonian water, I'd be dead. Well, I'd be very sick. Okay, points here, not just lettuce, orange juice, even though it's pasteurized. And we don't think about pasteurization, we think pasteurization milk, pasteurized orange juice you can look at. But orange juice can carry things. Again, it's how's it refrigerated? Are you walking by the salad bar where people are coughing on it and stuff? Look at the people with the gloves, right? You know what I mean? Food service. I mean, many people are very conscientious, but I often watch people, they put on gloves, and then they just start touching everything anyway. Well, okay, the bacteria that you might have had on your fingers isn't going in the food, but you just grab this, this garbage can and just, I'm not hungry anymore. I'll go cook something. So keep an idea what's going on. Okay, seminal typhoid fever. Oftentimes you can just let this one go. You can treat it, but this type of food sickness, typhoid lives only in humans. And we talked about the famous case of a food carrier in this typhoid Mary story. The thing about the carrier, you don't know that they're sick. Like I said, we're carrying different things. Not all of us the same thing. In typhoid fever, that is the case, and so you can transmit the disease, and it's harder to find. It's a harder case. Here's my thing: if you're traveling, boil it, cook it, peel it, or forget it. Use your best judgment. You know where you're going. If it's a nice place and people, but if you're in the bush, and you're watching piranhas eat catfish and stuff, and other things happening, you're going like, I'm pretty close to the food chain. I want to stay on this side of it. Then maybe you think about stuff. Okay, you get sick from typhoid fever, but it can get bad. You can actually die from it as it gets worse. my little commercial on Typhoid Mary, love talking about this. Here's a portrayal of Typhoid Mary in 1909. She's an Irish immigrant, Mary Malone, living in early New York, congested areas, immigration, you've seen the stories. It's not great living conditions, you're not making a lot of money, and you're not probably having central heating and all kinds of things. So there's a lot of disease, tuberculosis, all kinds of things. So that time in that area, here's the deal, she's cooking, and I don't know, she's putting some meatballs in the frying pan or something, I don't know. And this does come out. Okay, so she's being portrayed as Typhoid Mary. So right away, giving this nickname that captures fantasy, captures the concept of what it means to have germs and being spread in germs. Up until this time period, carrier-based disease transferal isn't that well thought through. This whole idea of not just social disease, but moving things through social circles and living conditions, usually the people they see that are hurting other people are the ones that are in the, are these people sitting in the beds and coughing up a lung or something. Okay, this kind of idea that you could be normal and a nice thing, top of the morning to you. That's kind of a different concept. Anyway, she's associated with carrier-borne disease, that type of root. We've talked about fecal-oral root. We've talked about the movement from fomite, an inanimate object. Fomite, like chewing on a pencil in lab. Okay, that's a fomite transferal. Okay, when I usually say take that out of your mouth, not being mean, it's like take it out of your mouth. It's a fomite transferal, and this is a carrier-borne transferal. Cook, she affected a lot of people. Some of them died. With her, they try to find her. They associate some sickness. Going, what's going on there? Maybe she's not washing her hands. I don't know what they were saying at the time. But she was hard to track down. Why not? You're trying to stay alive, just making them existent. She did die, but she died from a stroke. Never died from this disease, typhoid fever. She's associated with many cases of typhoid and deaths that could be recorded. Let's face it, late 1800s, early 1900s, we're not keeping records all that great yet. We actually didn't have these things at all. That kind of stuff. She was basically immune to it. Your immune system can develop immunity to different things, or just being sensitive to it. I got to end this part. I love this part. So Typhoid Mary probably didn't look like the lady coughing up the stuff on the meatballs. She looked more like this, and she captured a lot in history, not only because of her effect on other people, but because of this event that they're starting to associate how disease is transferred. Okay, I don't think Typhoid Mary woke up and going, I'm going to get electrocuted sick. I just think she was just this lady trying to do her job, stay alive, making it in an area that was probably marginally better than the area she immigrated away from. Don't know. I guess I'll have to read the book, but that's my guess. Nova gets the special. We got these mean cops trying to grab Typhoid Mary, and all of a sudden she's kind of transformed from perhaps a plainer-looking person to this fiery Irish-looking woman. I like that. I've never seen this movie. I Googled this and came up with Electra. Is this, anyone know, it's probably old now, I did this years ago. But, now Typhoid Mary is kind of good looking as a movie star-ish looking gal, okay? And she goes around apparently maybe kissing other women. I'm not sure how this is all working, I didn't see it, but basically the kiss of death idea. So how do we come up with false impressions of how the world works? I have no idea. It could not be Hollywood. So, in essence, Typhoid Mary didn't look like this or this. She probably looked a little bit more like me than them. And so, how you look is not the point. The point is, yeah, it's okay. Portray things through Outbreak. That's a good movie. Portray things. But realize, too, that sitting in the classroom listening to a professor yammer on about stuff, somewhere in there you're taking some of the facts away that you can process on your own. Shigella, I don't want to talk too much about Shigella. I mean, it comes up, it's a pathogenic to humans. You get Shigella, food poisoning, just know it's a proteobacteria. Yeah, like a lot of these enterics, you can move them around. Why are people always washing your hands? Why are you not touching the handrails as much as maybe you used to? Because people forget to wash their hands. Someone's doing a pretty creative exploration of where people might be moving around coli and stuff from bathrooms and other places in labs, so that's kind of fun. I found out, and if you look at the demographics, sorry to say this, men tend to not wash their hands as much as women. I'm not saying it's a sexist stat. Look at the stats at it, because the big hand washing campaign by ASM was a big deal. But grossly enough, more fecal contaminants were found in male keyboards than on female keyboards. So it's kind of like, really? So, can someone change the next slide for me? Anyway. Shigella, cramps, diarrhea, that's that kind of thing. How do you avoid most of these enteric things? We just said it. Wash your hands. Be aware of other people. It's not that soap kills this stuff so much. You think, oh, it's a gram negative. It should break them open. It's more so that action. You know, you guys say that, sing happy birthday, the ABCs. It's that action of moving off, using that hydrophobic surface to dissolve some of the oils on your hands that are holding on to those bacteria. That's it. Okay, not too much. Okay, some other things. Proteus, the Wanderer. We looked at Proteus in lab. If you're not in lab, what we saw is that these things are highly motile, very motile. Now, let me tell you about a few cool things and we get away from disease for a little bit, although Proteus can be bad. You can get somewhere in your urinary tract infection. Why? Who studied Proteus? What enzyme did they make that's diagnostic for this species? Anyone do that in lab? Urease. Why does urease come in handy in the urinary tract infections? Go figure. Okay, because it's a handy enzyme to have. It breaks down urea. Why do you want to break down urea? If you're a bacteria, what are you getting off of urea? A little bit of carbon, but more so what? Chemists, urea. You guys know this. You take an organic chemistry, you learn all this stuff. What is urea? Ammonia, yeah, so you get a good nitrogen source. Okay. Easy nitrogen source. Somewhat, a little bit more soluble in hydrophobic materials, so that they use urea to break down oil spills as a nitrogen source. And they use the hydrophobic phosphorus to go along with it. Clever, clever, clever. People are thinking about bacteria in the earth. Urea is a big thing. You have a source of urea, and they can be an opportunist and take care of it. Now, let's talk about something else. Yes, they have flagella. We know all about how that works. Look at this. Now, if we were to see this on a toadstool or some kind of growth in the forest, we go, oh, there's rings, and it's growing in rings, and that's happening because it's a fungus. Okay. This is probably a colony that's, let's just say it's 72 overs. I think it's a reasonable guess. Some of you may have seen this. Sometimes some strains of Proteus will make these waves. Oh, that's just the way they do it. Some make slimy waves. Some, like Bacillus, look kind of waxy, and they just all have a different colony morphology. Yeah, they do, but the wave thing is kind of neat. What they do is they'll grow, and they actually have cell-to-cell receptors. They tend to be a compound called a group of compounds, homoserine lactone. So a sugar with a amino acid configuration. And they seem to put out these things to say, slow down, come on, hang on. Okay. They're having a multicellular interaction. Now, when you think about bacteria, you tend to think that E. coli looks like that E. coli looks like that E. coli. Oh, that strain's making me sick, but it looks like that E. coli. You get the idea. Now, the idea that they can put out signal and go like, okay, slow it up. We're starting to out-eat our food source. That's what I think they're saying. Because you start here. This is the oldest part of the colony. They're growing out. They're spreading out like some of you are doing amoeba from the dirt. I need more food. Okay, wait a minute. We're growing a little bit too fast. Let's just slow down here. Take it easy. Okay, how's it going, Charlie? All right, let's go. And you grow it again. It's kind of slowing up because we're growing and eating up food faster. Now, that's my interpretation, but the signal is in there. Read Shimkitt's the multicellularity of single-cell life out of the University of Georgia. Great, great book, written around around 2000. But still, this idea that you can have swarming, okay, these things will actually move around each other because they're moral, but also signal, stop, and go. You see it also with bacillus. A lot of bacillus in lab, you see, oh, it's kind of waxy, okay, not too, every once in a while you see those strains that produce those feathers. You know, they look like frost on glass on a cold morning. You know, that's chaos theory, right? That's fractal movement of ice crystallization on a glass surface. These guys are also talking to each other and setting up these things, and they think that it can be some of the same thing of intercellular communication. This is crazy. Okay, you have intercellular communication, hormones, nerves, all kinds of stuff that's just talking to you all the time, chemicals and so forth. Bacteria, don't they just eat stuff and poop out stuff and get us sick once in a while? This is nuts stuff. So I'd like to talk about Proteus. I shall move on. Yeah, how much important in nature? Yeah, you know, growing your source. Let's just take it easy. I also think, and you just write this down. So 20 years from now, when I'm gone, and you guys read in the newspaper, bacteria move through the soil and attack people. Lemke was right. They move, I think, a lot more than we're doing. We just don't have the way of taking them. That's practical. We can't take them. Okay, some of the 2,3, that was a mixed acid. 2,3-butane guys. We in lab we know that there's a test to look for acetone. Okay, enough of this stuff. Kent State, they do some nice stuff. Here's what I was talking about. This is an Entero strip, API strip 20E for Enterix, 20 tests. And all those things we did in lab, I guess if you're not in lab, come on by. But we have all these test tubes, all these Petri plates, like, and Jessica and I are pour stuff DNA. Wait a minute, I can put all that stuff, this is the scale. These things are the size of like your pencil eraser, and you go through, you inoculate it, and you look for color changes. Same principle. I like test tubes because A, we can afford it. B, you can see things much easier, and as a learning tool, it works. Same principle. Go to the hospital, you'll probably be doing these. You can just score these at the bottom and look it up on a scorecard and get your species. Enterobacter aerogenes, I'm going to ask that species. We only have a few actually in these groups. Some can be pathogenic. They are certainly ones that are enterics, but they give a different reaction on EMB plates and a little bit different. And they're not like E. coli, they're 2,3-butane. Klebsiella pneumonia. Okay. Klebsiella lives in nature. All these things do. You can find Enterobacter in soil. You don't have to go into a cow's hinder to get some of this stuff. Klebsiella is the same way. The pneumonia part of Klebsiella is that it's pasty. In lab, the reason we did capsules, we grew this stuff in pasty old milk. Okay, and milk made this capsule. That capsule, that Klebsiella is in your lung, sticks to it. That makes it more pathogenic than ones that don't stick to it. As a postdoc, I was trying to study the extracellular matrix in nature as a food source for bacteria and algae. Enough on that. Someday if you want to talk about it, I would be happy to. But I found a person in Brooklyn who was trying to take off the extracellular matrix of Klebsiella pneumonia. He was interested in making people less sick. He was working in one of the densest areas in our country. He saw a lot of pneumonia. I was interested in taking that material as a labile carbon source and seeing if nature grew on it. The perspectives, same objective. Serratia, we know about serratia. We used to think it's a pretty common one. Some of you guys, I think, wrote about the experiment where they wanted to see how bacteria move through the atmosphere. and to study disease, so that they drop serratia from airplanes onto people. Did anyone, is that someone in this class? Do I have that about right?
Yeah, we had some.
You guys did, yeah, it's in your report, yeah. So, and you're like, oh, what were they thinking? Well, sure, if you grow these things up at 25 degrees, you can see the pigments, so they can go around and culture them. What a great idea. Whoops, these things actually make people sick. People got, what did they get, pneumonia? Did they get any gastrointestinitis?
I read urinary tract.
Urinary tract, urinary tract, yeah. So it's like, what a great idea. Man, maybe someone should have asked a microbiologist before you dump a bunch of bacteria out of a airplane. No one asked us. Okay, so anyway, red pigment, a pigment name that's very hard to pronounce, prodigicins, and really, it's a great pigment. It won't grow very much at 3725. It can vary from light pink to because there's several pigments in that cluster. So actually, if you want to study like the effects of UV or maybe some kind of mutagen, you can use serratia as one of your things, because you can knock out that pigment-forming thing before you kill them. We used to do an experiment with UV light with them. So here they are. They start to develop the pigment. You see it's different from coli and luteus. So we all like serratia because it's colorful. Okay. But it can cause a lot of problems. What do we got here? We want to get gamma, mix that. Okay, we talked about Proteus. We talked about Pseudomonas. We talked about Burkholderia. I think we'll just end with this one on Pseudomonas. Of the metabolically diverse bacteria, which there's many, most are in the proteobacteria. Probably the most metabolically diverse are the Pseudomonads, and some of those have been reclassified as Burkholderia. We talked about that. In this case, Pseudomonas is oxidase positive. It loves breaking down stuff and using oxidases. It's almost like bacterial bleach in some ways. Not technically, but it likes to break stuff down. So for breaking down oil droplets, for breaking down aliphatic and simple aromatics, it's a great one for bioremediation. See you this afternoon if you're in lab. Otherwise, see you on Thursday if you're not, or stop by. We'll talk as you need.